welcome back to episode 19 of Interview with Scientists series. Today we have Hamza. Hamza is a Toronto based bioinformatician and CTO co founder of BioBox Analytics. He did his MSc from Toronto, and during his MSc, he studied medulloblastoma using next generation sequencing and bioinformatic techniques. Today, we will talk with Hamza about BioBox, sequencing, and data analysis. Thank you, Hamza, for joining us and welcome to the show.
Perfect. Thanks for having me, Chinmaya.
Our pleasure. So let's go back to your background and can you tell us about your MSc work? Medulloblastoma sounds really fascinating. So what was it
Of course.
about?
Of course. So my undergraduate degree was actually in biomedical engineering. The focus was actually in robotics and lasers. So the lab was based out of Sunnybrook Hospital. So I got a lot of real-time experience in the actual hospital,
Okay.
specifically operating rooms. Near
Yeah.
the end of my undergrad degree, though, this opportunity came up with Dr. Michael Taylor at the Hospital for Sick Children.
Okay.
Um, and the reason his lab is so unique is he has the largest cohort of brain tumor samples, specifically medulloblastoma, in the world.
Okay.
He's got thousands upon thousands of them. Now, medulloblastoma is actually one of the most abundant malignant pediatric brain cancers
Okay.
um, that occur annually. I think the rate is about 300 children every single year, and
Wow.
primarily they affect children.
Mm-hmm.
What his lab has actually done is stratify these brain cancers, because whenever we talk about cancer, we
Yeah.
talk about it as like one disease, but it's actually unique sets. So one way of subdividing them is what type of cancer is it? Medulloblastoma, GBM, glioblastoma, so on and so forth. What his lab had actually done is looked at medulloblastoma, looked at it from different perspectives, and using sequencing technology actually determined that there are four specific individual types of medulloblastoma,
Mm-hmm.
each with their own unique signature. So
Okay.
Wnt, sonic hedgehog, group 3 and group 4. Wnt and sonic hedgehog is actually the pathways that are affected. And that only comes about by looking at different layers of data, specifically sequencing data. Um, so my time in his lab during the course of my MSc, I was fortunate enough to be trained by two fantastic postdocs who are now professors, Dr. Sorana Moresi and Dr. Florence Cavalli. Sorana is now at UBC, and
Okay.
Florence is in Curie Institute in France. And they both taught me multi-omic data from the ground up. The lab had a lot of different sequencing types.
Yeah.
So they taught me how to look at RNA, whole genome, whole exome, different types of analysis within each of these. So looking at structural variations, single nucleotide variations, expression, microRNA expression, circular RNAs, methylation. So it was a very robust amount of training. And then my specific specialty in master's was in long read sequencing technology, which had
Okay.
first come out.
Yep.
And then as the years progressed, single cell technology also came out.
Yes.
So I got trained on single cell as well as these synthetic, you can use single cell hybrid technology to try
Yep.
and reconstruct the genome using long and short reads together.
Yep.
Um, so I even got training on that. So it was really interesting to see just how much sequencing itself has proliferated in the field of biology, how much nuance and technique is involved,
Yep.
especially when you're learning what you have to look out for,
Yes.
and just the wealth of information you can get from that because it lets you analyze the disease in multiple layers. So while I
Yes.
was there, one of the actionable things from these multi-omic data
Yep.
was the four subgroups were actually subdivided into 12 subtypes. The
Okay.
reason these are significant is not just because we're just, I forgot what the term is, but you're splitting hairs.
Yes.
You're not splitting hairs for the sake of splitting hairs. When you split these according to these different signatures, you actually get distinct clinical signatures,
Okay.
and that helps guide treatment, especially when you're a parents or the child themselves, and you're trying to figure out whether the type of sonic hedgehog tumor you have gives you a year, gives you 3 months, or
Okay.
gives you a much better prognosis than that.
Wow, that's a lot of omics data that I've just heard, and that blew my
Yeah,
mind.
I can imagine. Yeah, their lab has a wealth of information.
Information. Correct. So, what was one critical issue you faced during your master studies, especially related to sequencing?
So there were a couple that I personally primarily felt. The first was because we had so much data itself, trying to organize the data as well as keep tabs on where data is becomes difficult. And the reason for this stems from the way a lab runs.
Okay.
Labs typically have, especially when you get to that size,
Yep.
labs have biologists who are trained and experts at extracting the relevant material, DNA, RNA, protein, whatever it might be,
Yep.
from the tissue that gets collected from the OR, preparing it, doing whatever they need to it, and then sending it for sequencing. Once the sequencing reads come back,
Yep.
all it is just a ambiguous file. And what I'd often get is, hey, let me forward you an email. There's some sequencing data there. Can you just import it into our shared cluster?
Okay.
Okay, perfect. I'll do that. 3 months, a year, 2 years from now, someone will ask me, hey, we found this data. Do you know what it is? Honestly, I don't. I'm not the one who ran the experiment. None of that is encapsulated within that system. So this broken telephone was a
Yep.
big hurdle when you're trying to track down specific data, tagging to specific phenotypes, to specific samples, whatever it might be.
Okay.
So that was one big disadvantage or hurdle that I had to overcome, which is how do I get all the relevant material that I need for both my masters as well as other people's projects that I'm supporting.
Yes.
The second was because there's this disconnect between expertise in each of these fields. Like I'm not a biology expert at all. I think the only thing I got to, or the most advanced thing I got to, was running PCRs and gel electrophoresis, and that was like, oh, this is perfect. Like I shouldn't go any further because that has its entire own endeavor. If I was doing a PhD, that might be different. So you have all these biologists as well that now rely on me to do the informatics analysis for their project. So I'm doing all these deep analytical perspectives on why something might be coming out a certain way, or just taking it all the way from the raw files to the results and letting them put in their expertise. While that works for one major project or a few major projects, it becomes a huge time sink. Like 90 to 95% of your time gets sunk into this when all you're doing is just going back and forth where the analysis isn't exactly what the person wanted, because unfortunately the biologists don't have the expertise to run the analysis themselves. And if it's not just the expertise, it's the resources. We have a shared cluster on our computer or at our lab, but because it has all of our data and it has everything, it is a bit of a security risk to just let anyone on there, because there could potentially run a command that overwrites certain data that we can't get back. It deletes the data, it moves things to a place that we don't know, or there's just no management as well. So trying to accommodate all of the requests that kept coming in internally as well as externally to the lab was a huge pain point that I had to, one, identify and realize, hey, this is a huge time sink for me, and then two, try and leverage what would be the best way through this process.
Okay, that sounds a bit complicated thing, which I am not personally aware of. But
Yes.
let's come to sequencing and qPCR data analysis. What is the difference between them? Because these are being commonly used right now during this pandemic.
Yes. So there's a key difference between them, right? So typical experiments. I'll start with PCR, for example. So PCR is polymerase chain reaction, and all it is you take DNA that you want, you target. So you design these primers, and primers are basically small pieces of DNA, forward and reverse, to target a specific section of DNA that you want to capture. You end up tagging it. You run PCR, which is a very common process that you can Google online. And all it does is it amplifies that region of DNA that you're interested in. So you go from 2 copies to billions of copies within the span of a few hours. You then run it through a gel called gel electrophoresis. And because DNA it can be ionically charged, and the length of your DNA can give you different voltages and different potentials, when you run it through a potential difference, the DNA fragments actually can get separated. And then when you have your target band that you want, you splice that out, you get rid of the gel, now you have your original DNA. And what people do is they'll take that and they'll send it for Sanger sequencing. So Sanger sequencing is the traditional way of sequencing, right? So what you do is you take your DNA, you take fluorescent nucleotides, and you let the replication cycle of DNA happen one base at a time. Every time a base happens, you have these high-powered cameras that just capture what wavelength came out. A, T, C, and G all have their own wavelength. And so you get this nice picture that goes through. So that's PCR and Sanger sequencing. when we talk about sequencing nowadays, we talk about next generation sequencing,
Yes.
which is taking this process of Sanger sequencing,
Yep.
but instead of shearing one specific piece of DNA, you actually shear the entire DNA, the entire transcriptome, whatever it might be.
Okay.
You end up embedding them at the bottom of these. So each of these instruments, Illumina is the one that I'm going to talk about because it's the most abundant.
Yep.
They have a plate on the bottom where each of these fragments of DNA or RNA or genomic material can actually ligate onto.
Yes.
They do PCR right at that layer, well, after it's tagged, so you get multiple copies, clusters of this specific fragment.
Yes.
Then from here, the machine, the next generation sequencing machine, basically does something equivalent of Sanger sequencing.
Okay.
There's a high-powered camera at the top, and it actually looks at a 2D picture, and it can tag cluster, position,
Yes.
with what signals are coming out of it.
Okay.
And so you're able to read multiple copies of DNA, RNA, whatever it might be, in real time, rather than each segment of that you would get from Sanger sequencing.
Okay, that's awesome. Thank you for the nice explanation. I always wondered about this difference between sequencing and PCR. So, what led to you? What led your team and you to start BioBox?
So the same hurdles that I was experiencing from my perspective as a bioinformatician, my co-founders Chris and Julian were experiencing it from their own perspective. So Chris and I worked on the exact same floor together on Circuits.
Okay.
It was in a different lab, kind of a different environment, but he was more biology focused.
Okay.
And he's a self-taught bioinformatician. So he would experience the hurdles that I experienced, but more from a biologist point of view.
Okay.
Julian worked in high performance computing, so in a cluster system. He experienced it from a data architecture perspective because he would also see the other kinds of requests that people would end up facing.
Okay.
So Chris and Julian knew each other through U of T. I knew Chris, and then I met Julian as well.
Okay.
And one day we're just sitting outside, we're just having a chat, and we're complaining about all these struggles that we're facing, and we're like, there has to be a better way. There has to be a better way. And then the idea of BioBox started forming through because instead of thinking about it from there has to be a better way, somebody else should be out there making it.
Yes.
We decided, hey, we firsthand know this problem. Why don't we go about solving this? And that's basically the idea of how BioBox came about. So the initial concept was, let's solve the problems that biologists, bioinformaticians, the field of biology is facing in the advent of next generation sequencing data becoming so thoroughly available. But let's solve it at all levels that may be experienced.
Okay. Awesome. Thank you for taking the initiative because, as you said, someone has to do it and you guys stood up.
Exactly.
So, how is BioBox going to solve the data analysis problem?
So, when we looked at the market at that point, because our first instinct is, hey, what else is out there that might already have been solving this? While there are great companies out there, each one is only solving one of 4 different kinds of problems. So when it comes to data analysis, what you need to do is you need to process the data, you need to visualize it, you need to analyze it, and then you need to explore it to really capture what people are doing with sequencing data or just data in general. Like I said, some companies will address one of the 4. Some companies are fantastic at processing data. They'll get you from raw sequencing to results, and then that's about it. But that is only part of the way of the full journey that has to be taken. What BioBox is trying to do is we're trying to consolidate the visualization, the processing, the analysis, and the further exploration of genomic data. So what we end up doing is we can take your raw data that you might have processed from a sequence experiment.
Yes.
We help make the actual processing itself really easy by providing pre-built workflows based on what are typically used as gold standards. Once you get your results, instead of just giving you this flat file, what we try and do is we help you with visualization tools as well. We help you actually interpret what this data is. Because of the wealth of knowledge out there, especially from large consortiums, studying these pathways, studying typical effects in standard data sets, we link your results with respect to everybody else's results. So you get pathway enrichment rather than having to export your data from one program and then embed it in another or embed it into a different browser. Or just literally go up to a list and ask people, hey, have you ever seen these genes before, right? Like, makes it a lot easier. What we also do is if I run an experiment on some project of mine where I've tagged all of my samples
Mm-hmm.
and everybody knows what it is, and a colleague of mine who is in my lab as well in the same organization
Yeah.
is just digging into his or her own data or asking a question through our knowledge base.
Yeah.
My results will actually come up to them. So what we're
Okay.
also leveraging is uh, the disconnect, or we're trying to overcome the disconnect in communication
Mm-hmm.
when it comes to personal projects, right? If
Okay.
you're collaborating with 10 people and they're all collaborating with each other,
Yeah.
each of your expertise will make every other project that much better.
Okay.
And that's how BioBox is trying to overcome the other four problems that exist within biology rather than just tackle them one at a time.
Okay.
Yeah.
Awesome. So I have seen the word pipeline and library on your website used quite frequently. So
Yeah.
what are these? What is a pipeline and a library?
So the library is our solution to that fragmented data problem that I was talking about before. Our library is a free-form um, utility where
Okay.
you can catalog any kind of samples, any kind of, we call them entities to
Mm-hmm.
be a more generic term,
Yeah.
but any kind of entities that may exist within your lab. So most labs will typically have mouse models.
Yeah.
They'll have progeny from those mouse models. They'll
Yes.
have patient xenografts that they've embedded within a mouse model. They'll have raw tissue,
Yeah.
so on and so forth. These right now, I could almost place a bet right now and say 90% of these samples are tracked in Excel sheets
Mm-hmm.
on people's local computers.
Yes.
It is completely illogical to do it this way, especially because, and the way my supervisor presented to me, biology and science is a revolving door. People
Yes.
come in, people go out all the time. So you need a solution that is long lasting and will be able to translate the information so that the field, the lab, can continue to survive. The library lets you design any kind of model, so
Yes.
however you're tracking it with whatever fields you want, and then add new entries throughout. You can compare and tag how entries relate to each other as well. And this
Yes.
includes when you do an experiment, let's say I take a sample,
Yeah.
I run it for sequencing, I get my data back. When I get my data back and you import it into our system, you actually specify what sample that belongs to. So nobody has to wonder where the file exists. We
Yes.
take all the complexity of that away, and all you have to ask is, for this sample, do I have data? What
Okay.
kind of data do I have? And we provide. So
Okay.
that's the library in a nutshell.
Okay.
The pipelines, so when I'm talking about processing raw sequencing data to a result,
Yeah.
that's what our pipeline studio is. So
Okay.
other companies have tried to do this as well, and they've been successful at it as well. They, the way you process raw data to a processed file is you string together bioinformatic workflows.
Mm-hmm.
The process of stringing this together
Yes.
is simplified within our system because we take the complexity away from the outputs from one workflow tool
Yes.
being compatible with the inputs of another workflow tool. A
Okay.
lot of it is data manipulation. We
Yeah.
make that simpler. We also so the way we approach our pipelines as well is we capture what version of every single tool we've installed.
Mm-hmm.
And when you execute it to process a file, we
Yeah.
capture all the parameters that were put into it.
Okay.
Science as a whole thrives on reproducibility
Mm-hmm.
because it improves transparency. That's the only way we can guarantee that specific outcomes, specific results we see in these papers
Yes.
are genuinely true. If I can reconstruct your experiment.
Yeah.
Right now, the standard for papers, especially when it comes to bioinformatics, is you'll see them say what tools they use, and
Yes.
sometimes they'll mention what parameters they used as well. But there have been other studies that have proven, depending on what OS you're running
Mm-hmm.
and what other libraries are installed, the math can slightly be different. We're dealing
Okay.
with billions upon billions of bases being aligned.
Yes.
Small differences can actually make a statistical difference down the line.
Okay.
So we end up capturing all of that information so that if you 5 years from now, 10 years from now, want to run and get the same results, you
Mm-hmm.
can take the same input files that we have, the
Yes.
exact same version of OS and tools, and get to your outputs that much faster.
Nice. So, BioBox is a cloud-based platform. So, how safe is the data in the cloud?
It is extremely safe. The reason for this is a lot of these large consortiums,
Yeah.
including government facilities, have started moving their data to the cloud
Mm-hmm.
because of how much more robust and protected it can be. Data centers themselves have maximal levels of security, restricted to only their personnel, and even within only their personnel, you need the specific clearance, and they get audited multiple times.
Yes.
Google also releases and prides themselves on it on being HIPAA compliant
Yeah.
because of how secure their servers are. Beyond that, we at BioBox take great care at going even further than that by anonymizing all data that is kept at rest as well
Okay.
as in transit.
Okay.
And then because the data files themselves aren't directly correlated with the samples when you're uploading them, everything is
Yes.
anonymized even further. So
Okay.
even if your data was anonymized from the start,
Yeah.
we try and add more layers of complexity or anonymity so that there is a more personal guarantee that everything is nice, safe, and
Safe.
secure.
Okay. That
Yeah.
sounds soothing to the ears. So, can we analyze qPCR data on BioBox as well, or just sequencing data?
So qPCR data you don't typically analyze on any kind of platform. qPCR data comes back, and essentially all it is you just look at peaks and bands
Yes.
on a specific position.
Yeah.
So currently on BioBox, we haven't made that utility yet, where you can just observe a common strip.
Yes.
You can, however, process and observe RNA sequencing data,
Yeah.
especially paired end sequencing data to get expression counts. You can do whole genome sequencing data and whole
Okay.
exome sequencing data to get copy number information, single
Yes.
nucleotide variation. And coming up soon, we will also do, I believe we're going to expand it into methylation and single cell sequencing,
Okay.
but that's in
Okay.
the coming quarters.
Okay. Awesome. So, do will we have a desktop version of it sometime?
So we've debated the idea of having a desktop version, a utility that you can just install and then
Yes.
just run it through. And we initially in our prototyping phases, we did actually create one, but there are some pros and cons to it. So the pro is you have everything running locally,
Yeah.
which people just feel more comfortable with.
Yes.
But the con and the major con I would say is security.
Yes.
Because it's running on your local system, but you're tapping into external data, it's far better to keep that external data external and never access your system. That's one.
Yeah.
Two, if it is local on your computer and your computer gets lost, it becomes more of a headache, right?
Yes.
But the major one is we update our system almost bi-weekly,
Okay.
where the second we get new features ready that can help users, we try and push them out. And we want like letting users know rather than deal with software updates every single 2 weeks, which I
Okay.
also know people hate because I personally have mine turned off.
Yes.
Having it be a cloud-based system and through the browser allows users to have this seamless experience where every time you log in, you have the latest version of BioBox that we have released.
Okay.
And so it helps keep that flow going. If their new tool comes out, you don't have to worry about downloading it back again.
Yeah.
It's already going to be out there.
Okay. Thank you. So we had a lot of serious talk. So out of let's move on to non-serious talks.
Sure.
So what do you enjoy the most? Your hobbies?
My hobbies, I'd say, well, COVID's kind of changed what my hobbies
Of
are.
course.
Um, my hobbies typically include biking, especially nowadays. I've just gotten really into biking because the second the weather got better, I
Yeah.
just want to be outside as much as I can.
Yes.
Um, the other thing I'm just waiting on is soccer. Is a big hobby of mine to play. I
Yes.
haven't really been keeping up with the leagues that much, but the only sport I have been watching is the Formula One Grand Prix that have been happening. Like
Okay.
religiously, I watch them like 8 a.m., 9 a.m., whatever it might be. I'll be
Yes.
sitting there and going through it.
Nice. So will there be any job opportunities for at BioBox soon?
Yes, hopefully, not hopefully, there will definitely be expanding job opportunities in BioBox in the coming quarters.
Okay.
We're always on the lookout for fantastic talent as well.
Okay.
So if anyone's ever interested, they're more than welcome to send an email along. Um, we love everyone who works at BioBox. Like we love
Yes.
expanding our team.
Yes.
We love culture adds rather than culture fits because
Yeah.
you always want to expand yourself.
Yes.
But we'll be doing more rigorous hiring in the coming quarters.
Nice. So I would encourage my listeners and viewers of this video to watch out BioBox website.
Yeah, of course. And then your viewers and users can also, if they want to learn more about bioinformatics in general,
Yeah.
we actually have a blog post that we publish every week, every 2 weeks, where
Yes.
we've already gone over the basics of sequencing, the basics
Yes.
of whole genome, RNA sequencing, what they should and shouldn't look out for.
Yes.
And it's been instrumental in trying to educate the biologists to be
Yes.
able to be more independent.
Yeah. No, I have been following your blog posts, so I really enjoy reading it.
I'm glad to hear that. I'm glad people are actually getting value from it.
Yes, definitely. Thank you, Hamza, for speaking with us today. We encourage everyone who is watching this video or who is going to watch the replay to sign up on BioBox. Sign up for the BioBox, and we will be providing the link to that. Thank you very much.
Perfect. Thank you, Chinmaya. Thank you, everyone, for listening.